Hey, what is going on? It is March 14th, 2022, Monday, beginning of a new week. And I am beyond excited. Again, whenever I do these videos, I am beyond excited. But I do realize I'm kind of going through all of these videos and video that's fast, and I'm probably making a few mistakes along the way, which I totally get. It happens. And I plan to go back to these videos and redo them and hopefully correct my mistakes. It takes a while to kind of get the flow of these videos or do these videos. I mean, I have to kind of plan what I'm going to add to each of them, and I have to kind of like figure out what's the best thing to present at what time. And I realized that, yeah, there's so many levels that are running parallel, the higher level, the mid level, the low level in terms of detail, that like if I go through all the high level details, we might miss talking about some of the mid level details which are important for the next video, and which if you're looking at this video, we might gloss over by accident or get wrong, and you might be confused later on. But that's something I'll get to. For right now, I'm just making sure I built the foundation for the biochemistry I want to essentially be an expert in order to figure out how we can figure out a way that we can achieve eternal life. Okay. And that sounds pretty crazy. Now that I think about it, every time I talk about that, I really think I'm nuts. But I mean, really, honestly, if we could live forever in a healthy way, why wouldn't we? I mean, we could go into the philosophy of that, but I digress severely. So let's talk about. I've talked about yesterday. I left off at the Kreb cycle, and I didn't. I went through, I think, pretty much all the steps. Back up to oxaloacetate recycling and re-entry into the Krebs cycle. But there are a few things that I kind of glossed over, or specifically the function of the dehydrogenases, which is, I mean, that's kind of understood if you've looked at enzyme chemistry at all, and a few other things. Were the structures of a lot of these intermediates. Some of the things that are interesting, one thing I did talk about yesterday was the aspartate malate shuttle, which was important because some of these intermediates, they're not just confined to the mitochondria, they exit the mitochondrial membrane, the inner and outer membranes, and they, outside of the mitochondria, they recombine and are transported back into the mitochondria to be replenished for different phases of the cycle. So an interesting thing to consider, and something to wrap your head around, is that this cycle, and along with all the other complementary processes to ATP generation, are not running in parallel, not running in series with each other. They kind of run in parallel, and each step of this cycle is regenerated by the presence of the intermediate that may have been obtained from a different cycle, or a different instance of the cycle, or a different or from the aspartate malate shuttle, right? Like for example, succinate is not always coming from alpha ketoglutarate is not always coming from isocitrate carboxylation. It could be coming from the conversion, a conversion from one of the reagents in the that are brought in through that aspartate malate shuttle into the mitochondria, right? So you have your glutamate gets deaminated and converted to alpha ketoglutarate, right? So these reagents are regenerated in the cycle, and they affect the rate in equilibrium of this cycle, and thus the production of ATP. So this dance is very complicated because regulation of this cycle in nonlinear ways makes this complicated. So one thing I did want to talk about was let's dive a little deeper into some specifics and unique elements. So what you notice is that one thing I think is unique is this FAD to FADH2 reduction. Okay, so why do I think that? Because you don't really see this anywhere in glycolysis, or you don't see this in you don't see this directly interacting with the main uh, cyclic branch of this process in glycolysis in the transition phase or in the Krebs cycle. But FADH2 is important in the electron transport chain, which we'll get to, which is the mechanism by which the majority of the ATP is produced. And it is also a coenzyme that is involved in the control and activity of these in the enzymes like succinyl-CoA synthase, alpha ketoglutarate dehydrogenase. That FADH2's presence regulates these enzymes and their activity in converting alpha ketoglutarate, for example, to succinyl-CoA. Also, FADH is a regulator of the pyruvate dehydrogenase complex, which exists in the transition phase between glycolysis and the Krebs cycle. So we have to, it is indirectly, or it is tangentially interacting with these processes in order to regulate it, but and directly interacting in the electron transport chain, but it is also directly interacting, it's directly essentially being reduced here. FADH gets reduced to FADH2. So what is FADH? I'll just go into a quick spiel on that. So FADH comes from, it's the FADH2 flavin adenine dinucleotide. Let's just talk about the structure of FAD, right? Is this molecule which is derived from riboflavin. Okay, flavin means yellow in Latin. Riboflavin is vitamin B2. So if I look at vitamin B2, riboflavin, right, you'll see that riboflavin looks like this. Okay, so that is the core element, which is the coenzyme, which is structured as a coenzyme, so it can assist other enzymes, as I talked about, in doing their activity. And how does it do that? So this is the riboflavin consists of the ribose sugar, right? This is a sugar, right, with all these OH groups attached to this carbon chain. If you remember your definition of sugars, this is in a three-ring heterocyclic moiety, right? I think it's called isoxyloadenine, but and it's especially this part right here is the important part of this three-ring moiety, because this is the reactive part, which is implicated in the reduction of FADH. So it's more of a complicated chemical process that occurs, but we won't go into the chemistry there. I just wanted to kind of go over FADH and know that the chemistry exists. If you actually want to go into the chemistry, know that flavin, there's actors called, there's flavin adenine dinucleotide, and then there's flavin mononucleotide. And just to quickly go over the difference, right, flavin adenine dinucleotide, right, has this adenine nucleotide attached, right? Flavin is, I think, sometimes called a false nucleotide, but it's attached via this pyrophosphate, or this 2-phosphate linkage, to this nucleotide, adenine nucleotide group here, which consists of the adenine nucleotide and the adenosine amino acid, or the adenine amino acid. Then that is connected to this reduced sugar, which is again connected to the flavin, this heterocyclic moiety, which is this riboflavin section here. So, and essentially, again, this is important in the electron transport chain, and I just wanted to quickly note that, again, it's different than NADH. It's actually used as I've seen it. It comes from milk, and it was one of the first coenzymes, that was really studied when the whole discipline of coenzyme activity came to fruition in the 1930s. So yeah, and you can get it from different like milk is one of the sources where it's also referred to as lactoflavin. And your body essentially doesn't produce it. You need to ingest this in order to continue on with this with the Krebs cycle. So it's an important an important endogenous substance, for a flavoprotein, which your body needs. So it's also involved in many other reactions in the body, but just because we're talking about its reduction reaction to, or double reduction reaction. Remember, it takes two protons and adds them, or two electrons, and adds them to itself through its interaction with the enzymes that reduce it. We have FADH2. So that's all I wanted to go in there. At some point, I'm going to go in deeper into FADH2. But for now, I just wanted to kind of go over why we would need it, since we do need it for the electron transport chain, and it's a co-carrier of the electrons along with NADH. NADH I'll go into in its own separate video. But for now, thanks for listening. I'll see you around. And I'll just quickly, to kind of attempt a little bit of a schedule for the next time, I'm going to talk about, next time I'm going to continue on with NADH, and then I'm going to continue on with the electron transport chain. Okay, yep, thanks a lot for listening. I'll see you around. Oh, and like, subscribe, and comment.